Welcome to the Deep Dive, the show that cuts through the noise and gets straight to the core of what you need to know. Have you ever really stopped to consider the sheer speed at which life unfolds?
It's kind of amazing when you do, isn't it?
Yeah, I mean, think about it. From the incredibly complex process of digesting your last meal, breaking it down, absorbing nutrients,
Mhm.
to the lightning-fast cascade of thoughts, emotions, memories firing in your brain right now, how does it all happen so rapidly, so precisely, so sort of effortlessly, moment after moment?
Well, it feels effortless, but underneath, there's this incredible molecular machinery working nonstop.
Right. It's not magic, though it often feels like it. And today we're diving deep into the microscopic maestros, the unsung heroes that make all that incredible speed and precision possible: enzymes.
The tiny powerhouses.
Exactly.
Yeah.
These biological catalysts are the hidden towerhouses, the molecular accelerators behind virtually every chemical reaction in your body. And honestly, truly understanding them is, feels like a shortcut, a fast track to understanding life itself. We're going to unpack the secrets of these tiny biological workhorses.
And that's our mission today, really, to demystify the fascinating yet often overlooked world of enzymes. We're going to explore their fundamental structure, dissect precisely how they manage to supercharge reactions by factors that are, well, frankly, mind-boggling.
Yeah.
We'll navigate their intricate classification system, how scientists keep track of them all,
Okay.
and uncover the clever, sophisticated ways our bodies control their activity, turning them on or just dialing them up and down as needed.
Right. That regulation piece is huge.
It is. And we'll also touch on why this knowledge isn't just academic curiosity. It's absolutely crucial in fields like medicine, diagnostics, drug development.
It's real-world impact.
Definitely. By the end of this deep dive, you'll undoubtedly walk away with a profound, maybe even awestruck appreciation for these molecular workhorses that underpin your very existence. What's truly fascinating here is just how foundational they are to every single biological process, from your first breath to your last thought.
And to guide us on this incredible journey, we haven't just skimmed the surface. We've pulled together a really comprehensive stack of sources.
Yep, we've got the good stuff.
We're drawing from foundational biochemistry texts, the heavy hitters like Blanco, Champy, Harper, Harvey, and Kuhlman, alongside some deeply insightful excerpts from Vesius. We've distilled the most important nuggets of knowledge from all of them.
Trying to get the core ideas across.
Exactly. To give you a truly well-rounded, accessible, and insightful look into the remarkable world of enzymes. So let's begin this journey to become well-informed on life's incredible molecular accelerators.
Let's do it.
So to truly appreciate their impact, let's start at the very beginning. What exactly are enzymes at their core? We hear the term tossed around, in nutrition and biology, but what's the fundamental definition?
Okay, at their most fundamental, enzymes are biological catalysts.
Catalysts. Okay, what does that actually mean?
Simply put, it means they accelerate chemical reactions. They speed them way, way up. But crucially, they do this without being consumed or permanently altered in the process.
Ah, so they're reusable.
Exactly. Think of them as tiny, tireless facilitators. They step in, speed things up, and then they're ready to go again for the next reaction.
And what are they made of, mostly?
Almost exclusively, enzymes are proteins. Intricate chains of amino acids folded into highly specific three-dimensional shapes.
Proteins. Okay, that makes sense.
But there's a fascinating little exception. It turns out some RNA molecules, known as ribozymes, also have catalytic activity.
Really? RNA?
Yeah. It's a compelling reminder that while proteins clearly dominate the world of biological catalysis, they aren't the only game in town when it comes to speeding up life's reactions. It's almost like RNA decided to moonlight as an enzyme.
Yeah.
Talk about multi-talented.
Huh. Biological catalysts, not consumed. Sounds powerful, but how powerful are we really talking here? Is it just a slight nudge, or is it something far more significant that actually underpins life?
Oh, it's far, far more significant. The power of enzymes is truly astounding. Almost miraculous, really. They can increase reaction rates by factors ranging from 1,000-fold, that's 103, to 100 million-fold.
100 million-fold.
100 million times faster.
And in some exceptional cases, even up to a staggering 10,017 times faster compared to the reaction just happening on its own.
10 to the 17th. And
Yeah.
I can barely comprehend that number.
It's mind-blowing. To put that in perspective, imagine a chemical reaction that would, without an enzyme, take millions of years to complete.
Okay, geological time.
With an enzyme, that same reaction could occur in mere milliseconds.
Milliseconds. millions of years down to millisecond.
Exactly. It's the difference between, yeah, a geological timescale and an instantaneous cellular event. Without them, life as we know it simply couldn't exist at the incredible speed and efficiency that it does.
So that speed is fundamental.
Absolutely. This immense acceleration is precisely what allows for the complex, rapid, and finely tuned metabolic processes that sustain every living cell, every organ, and ultimately you. It's how your body keeps the lights on, not just for an hour, but for your entire lifetime.
Right.
Wow. 1,017 times faster. That's truly mind-boggling. And one of the things that I find so incredible about enzymes is their amazing specificity. They don't just speed up any reaction, do they?
No, absolutely not. Specificity is not just a characteristic, it's a hallmark of enzymes, a core principle of their function.
Okay.
Each enzyme is exquisitely designed to interact with a very particular molecule called its substrate.
The thing it works on.
Exactly.
Yeah.
Or sometimes a very limited range of structurally similar substrates. And furthermore, it typically catalyzes only one specific type of chemical reaction.
So super focused.
Think of it like a highly specialized lock and key mechanism, but on a molecular scale.
Ah, the classic analogy.
Right. A specific key, the substrate fits only a specific lock, the enzyme's active site, to initiate a particular predetermined reaction.
And that precision is vital.
It's absolutely crucial. It ensures that in our incredibly complex biological systems, only the necessary reactions occur at the right time and place, preventing unwanted side reactions that could be, well, disastrous. It's a cellular masterpiece of targeted efficiency.
So they're not just fast, they're incredibly precise. And they accomplish all this under conditions that are, perfectly compatible with life.
Yeah.
Which is quite different from what we might see in an industrial setting, right?
Precisely.
Yeah.
Their efficiency isn't just about speed and specificity. It's profoundly about the conditions under which they operate.
Okay.
Enzymes function optimally under the mild physiological conditions found within our bodies, our normal body temperature, a very narrow range of pH, atmospheric pressure.
Just everyday conditions for us.
Exactly. And this is a stark, crucial contrast to industrial catalysts, which often demand extreme temperatures, pressures, or highly acidic or alkaline environments to achieve similar reaction rates.
Right, you can't run a blast furnace inside a cell.
You really can't. The ability of enzymes to work within the narrow, delicate parameters of living systems is fundamentally what makes complex, sustained life possible. It's a brilliant piece of biological evolution. Really, enzymes designed perfectly for where they need to work.
And it's not like they're just constantly on, running wild, speeding up everything without any control. Our bodies have ways to manage them, right? To keep them in check.
Yes, absolutely. Enzyme activity isn't a free-for-all. It's meticulously and dynamically regulated.
Dynamically regulated, okay.
Our bodies possess sophisticated mechanisms to fine-tune enzyme activity, either increasing or decreasing it based on the cell's immediate metabolic needs or the signals it receives.
So they respond to demand.
Exactly. This regulation can be instantaneous, responding to momentary changes, or more long-term, adjusting to broader physiological demands. It ensures that metabolic pathways are finely tuned, preventing wasteful overproduction or critical shortages,
Resource management.
and that cellular resources are used with peak efficiency. And this raises an important point. What happens when this intricate regulation goes awry?
Yeah, things must go wrong sometimes.
They do. The consequences can be severe, leading to various diseases and disorders, highlighting just how critical proper enzyme control is for maintaining overall health.
Okay, we've marveled at how these tiny workhorses operate and how our bodies keep them in check. But imagine the sheer number of different enzymes out there, it's the thousands. Without some kind of universal ID system, it would be utter chaos trying to talk about them, study them.
Oh, absolutely. Total confusion.
So how do scientists bring order to this vast microscopic workforce? How do we name and classify nature's workers?
You're right. With thousands of enzymes, a systematic approach is not just helpful, it's absolutely essential. Historically, enzymes often got common or trivial names.
Okay, like nicknames.
Sort of. These names usually end with a suffix "ase" and often give us a hint about their substrate or the type of reaction they catalyze.
Right, like lactase, dehydrogenase.
Exactly. Lactase breaks down lactose, dehydrogenase moves hydrogen atoms, urease acts on urea. Pretty intuitive sometimes.
But potentially ambiguous.
Yes, while these common names are easy to remember and still widely used, they can sometimes be ambiguous or not fully descriptive of the enzyme's exact function.
I can see that, especially if you have similar substrates or reactions. How do you avoid that ambiguity and ensure everyone, everywhere, is talking about the exact same enzyme?
That's where systematic nomenclature comes in, providing crucial clarity. The International Union of Biochemistry and Molecular Biology, or IUBMB.
IUBMB, got it.
They developed a comprehensive and universally accepted classification system specifically to address this potential confusion and ensure unambiguous identification for every known enzyme.
A universal standard.
Precisely. It assigns a unique EC number that stands for enzyme commission number to each enzyme.
EC number.
Yep. And it's essentially a four-part code. Each number tells you something specific. The enzyme's main class, its subclass, its sub-subclass, and finally its serial number within that really detailed category.
Wow, 4 levels deep.
It ensures absolute clarity across all scientific communication. So when you hear EC 1.1.1.27, you know exactly which enzyme we're talking about, no room for doubt.
That's clever. And necessary, I guess. So the IUBMB gives us 6 main classes, a kind of master categorization. Can you walk us through those? What are they and what exactly do they do?
Certainly. These 6 classes provide a clear, logical framework for understanding the incredible diversity of enzyme functions across all living systems. This classification really offers a mental shorthand, a way to instantly grasp an enzyme's core mission.
Okay, class 1.
First, we have oxidoreductases, EC1. As the name vividly suggests, these enzymes are the body's energy traffic cops, basically.
Energy traffic cops, I like that.
They catalyze oxidation reduction reactions, redox reactions, meaning they are responsible for the precise transfer of electrons or hydrogen atoms from one molecule to another. This is fundamental to cellular energy production and many other vital processes.
Okay, give me an example.
A classic example here is lactate dehydrogenase, or LDH.
LDH, okay.
LDH is vital because it converts lactate, which is a byproduct of intense muscle work or low oxygen conditions.
Right, you feel that burn.
Exactly. It converts that lactate into pyruvate, and at the same time, it transfers hydrogen atoms from the lactate to a molecule called NAD plus randerphide. This is a crucial step in managing energy production, especially when oxygen is low, allowing muscles to keep working. When you hear oxidoreductase, you instantly know we're talking about moving electrons or hydrogens around fundamental energy currency stuff.
Got it. EC1, redox reactions. What's next?
Next up are transferases, EC2. These enzymes specialize in catalyzing the transfer of functional groups.
Functional groups? What?
Think of them as molecular couriers, moving specific chemical parcels like methyl groups, alisyl groups, glycosyl groups, or phosphate groups from one molecule, the donor, to another, an acceptor. They're essential for building and modifying complex molecules.
Okay, an example for transferases.
An excellent example is hexokinase.
Yeah.
This enzyme transfers a phosphate group from ATP, the cell's energy molecule, onto glucose.
Ah, the first step in using sugar.
Exactly. It's the very first foundational step in glycolysis, the pathway that kicks off glucose metabolism for energy production. So, hexokinase is crucial for getting our cells powered up.
EC2, transferring groups. Makes sense. Number 3.
The third class is hydrolases, EC3. These enzymes are the molecular unzippers, or maybe scissors, of the biological world.
The scissor, okay. Cutting things apart.
Right. They catalyze the hydrolytic cleavage of various bonds, CO, CN, CS, PO bonds, by adding a molecule of water. They essentially use water to break down larger molecules into smaller, often more manageable components.
Like digestion.
Digestion is full of hydrolases, yes. A specific example is urease. It catalyzes the hydrolysis of urea into ammonia and carbon dioxide. This is essential in the nitrogen cycle in nature and also has industrial uses, like removing urea from wastewater.
Okay, EC3, using water to break bonds. What's EC4?
Fourth, we have lyases, EC4. These enzymes are quite interesting because they catalyze the cleavage of CC, CO, CN, or other bonds, but without using water like hydrolases do. They do it by elimination, often forming new double bonds in the process.
So breaking bonds without water.
Correct. And what's also unique about lyases is that these reactions typically do not require energy directly from ATP. Unlike some other bond-breaking or bond-forming enzymes, they essentially split molecules, but in a non-hydrolytic, non-oxidative way.
Example.
An example here is fructose 1,46-bisphosphate aldolase. It works during glycolysis, cleaving a 6-carbon sugar, fructose 1,6-bisphosphate, into 2 smaller 3-carbon molecules called triose phosphates. This is a critical step for continuing energy extraction from glucose.
EC4, breaking bonds without water or oxidation. Got it. Number 5.
Moving on to the 5th class, we find isomerases, EC5. These enzymes are molecular rearrangers. Yeah, they specialize in catalyzing intramolecular rearrangements, meaning they simply move atoms or groups around within a single molecule, converting it into an isomer, a molecule with the same atoms but arranged differently. They don't add or remove anything, they just change the structure.
Okay, like shuffling the deck chairs on the molecule.
That's a good way to put it. Triose-phosphate isomerase is a beautiful example. It interconverts the two different triose phosphates produced by aldolase, dihydroxyacetone phosphate, and D-glyceraldehyde 3-phosphate. This is crucial because only one of them can directly continue down the glycolysis pathway. The isomerase ensures both halves of the original sugar molecule can be used efficiently for energy, maximizing the yield.
Smart. EC5, rearranging atoms within a molecule. And the last one, EC6.
Finally, the sixth class is ligases, EC6. These enzymes are the joiners or molecular welders of the cellular world.
Putting things together.
Exactly. They catalyze the joining or ligation of two molecules to form a larger one. And this process almost always requires energy, which is usually supplied by the hydrolysis of ATP. Because they synthesize larger molecules, they're often called synthetases.
Makes sense. Joining needs energy. Example.
A very well-known example is DNA ligase. It painstakingly joins fragments of DNA strands together during DNA replication and repair. Absolutely crucial for maintaining the integrity of our genetic material.
Definitely vital.
Another one is glutamine synthetase, which forms the amino acid glutamine from glutamate and ammonia, consuming ATP in the process. This highlights their vital role in building complex molecules needed for structure, communication, storage, all sorts of things within cells.
EC6, joining molecules, usually using ATP. Wow, okay. 6 classes covering pretty much every type of chemical transformation needed for life. That classification system really helps put the sheer variety of enzymes into perspective. Oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases.
That's the big 6.
But beyond these categories, how do these enzymes actually do their work? What's the secret to their incredibly precise action, particularly within that special place you mentioned earlier, the active site?
Ah, yes, the active site. It is indeed where the magic happens. It's the very heart of an enzyme's function.
Describe it for us.
Okay, so it's a specific, intricately shaped region on the enzyme molecule, often described as a pocket or a cleft on the surface.
A little indentation.
Exactly. This pocket is meticulously formed by the precise three-dimensional folding of the enzyme's amino acid chain. It creates a unique microenvironment, perfectly tailored for the reaction.
And that's where the substrate goes.
That is the crucial area where the substrate binds and where the actual catalysis, the chemical transformation, occurs. The amino acid residues within this site are arranged in a very particular spatial orientation that's perfectly suited for interacting, even if just transiently, with the substrate.
So the substrate just slots into this active site. Is it a perfect static fit, like a piece of a puzzle, or is there more to the interaction than just passive binding?
Oh, it's far from passive. It's a very dynamic, active interaction. When the substrate binds to the active site, it forms a temporary complex called the enzyme-substrate complex, or ES complex.
ES complex, okay.
And this binding often isn't just a simple docking, like fitting into a rigid slot. It frequently induces a subtle but significant conformational change in the enzyme itself.
The enzyme changes shape.
Slightly, yes. And sometimes even the substrate changes shape a bit upon binding. This dynamic reshaping, where the enzyme subtly molds itself around the substrate, is what we call the induced fit.
Induced fit, not just lock and key then.
Not purely. This induced fit process facilitates the catalytic event. It brings the reacting groups into the ideal position, creates the optimal electronic and physical environment for the reaction to proceed much faster. It's more like a flexible glove molding perfectly to a hand, rather than a rigid key fitting a rigid lock.
And this dynamic interaction is the core mechanism, right? How enzymes make reactions happen so much faster. They drastically lower the activation energy. Can you explain that in more detail? Maybe using our mountain tasks analogy again?
Yes, this is the absolute essence of enzymatic catalysis, the fundamental reason for their incredible speed. Enzymes provide an alternate reaction pathway with a significantly lower activation energy.
Activation energy, the energy needed to get the reaction started.
Precisely. We denote it as Ag, delta G, double dagger. Crucially, enzymes do not change the overall free energy difference, or GUG, between the reactants, starting materials, and the products, what you end up with.
So the overall energy change is the same.
Exactly. Nor do they shift the equilibrium of the reaction, how much product you get eventually compared to reactant. What they do is dramatically speed up the rate at which that equilibrium is reached. They just make it easier and faster to get there.
Okay, so back to the mountain pass. The enzyme doesn't change the height difference between the starting valley and the destination valley.
Correct, the overall Ag.
But the normal path involves climbing over a really high mountain peak that's the activation energy.
That's the uncatalyzed reaction, yes. Yes,
The enzyme essentially builds a tunnel through the mountain or finds a much lower pass.
that's a perfect way to visualize it. It opens up a lower, easier path over the pass, representing that high-energy transition state. This makes it much, much faster to get from one side, reactants, to the other products, even if the start and end points remain the same height.
How do they actually create that lower pass? What's the trick?
The key mechanism is called transition state stabilization.
Transition state. That sounds unstable.
It is. The transition state is a fleeting, unstable, high-energy intermediate form that the substrate must pass through as it transforms into a product. It's the very peak of that energy mountain.
Okay.
Enzymes are exquisitely designed to bind and stabilize this transition state more effectively and with greater affinity than they bind the actual substrate itself.
So they grab onto that unstable intermediate really tightly.
Exactly. By doing so, they drastically reduce the energy barrier required for the reaction to proceed, allowing it to occur much more rapidly. This stabilization essentially makes the peak of the energy mountain much lower and easier to get over.
That makes sense. So we mentioned the lock and key model earlier.
Right. Fisher's model from the late 1800s.
It proposed a rigid active site perfectly complementary to the substrate. Simple, intuitive.
Very influential, and it explained specificity beautifully. The idea was only a substrate with the exact shape could bind.
But then came this induced fit idea you mentioned.
Yes, from Daniel Koshland. It's a more refined and now widely accepted model. It suggests the active site isn't rigid, it's dynamic and flexible.
The glove molding to the hand.
Exactly. Substrate binding actually induces that conformational change in the enzyme, a subtle reshaping, and sometimes in the substrate too, to achieve the optimal fit and maximum catalytic efficiency.
So the interaction itself perfects the fit.
Right. This dynamic interaction ensures that the amino acid residues in the active site are precisely positioned spatially to maximize the catalytic process. It helps explain how enzymes achieve such remarkable rates and specificity, truly adapting to their molecular partners.
Are there specific chemical tricks they use within that active site?
Oh, absolutely. Beyond the binding models, enzymes employ specific catalytic strategies, for instance, acid-base catalysis.
Okay.
Amino acid residues in the active site can act as highly localized proton donors, acids or acceptors, bases, facilitating crucial proton transfer steps that are often part of reaction mechanisms.
Like passing protons around.
Precisely. Then there's covalent catalysis.
Forming a bond.
Yes, a transient covalent bond forms between the enzyme and the substrate. This creates a temporary, often more reactive intermediate, which then quickly breaks down to release the product and regenerate the free enzyme.
Okay, what else?
They also masterfully utilize proximity and orientation effects.
Bringing things together.
Exactly. Enzymes literally grab substrates and hold them close together in the perfect alignment for reaction. This dramatically increases their effective concentration right there in the active site and boosts the likelihood of a productive collision.
Makes sense. Anything else?
One more common strategy is inducing substrate strain or distortion.
Straining the substrate, like bending it.
Sort of. The enzyme can bind the substrate in a way that slightly distorts its shape, perhaps twisting or stretching certain bonds, making it more closely resemble the high-energy transition state.
Pushing it towards reacting.
Exactly. Imagine bending a stubborn twig, just enough to make it easier to snap. Enzymes do this at a molecular level, essentially giving the reaction a physical nudge towards the transition state. These intricate mechanisms really show the sophisticated, tailored chemistry happening inside that tiny active site.
So enzymes are these incredibly complex protein structures with precise active sites that can even reshape themselves for optimal function. But do they ever need a little extra help to do their job? A kind of supporting cast? They don't always fly solo, do they?
That's a great question. And no, they often don't fly solo. While the protein part is absolutely crucial, many enzymes require additional non-protein components to be fully active and do their job properly.
Okay.
These essential helpers are collectively called cofactors.
Cofactors.
Without their specific cofactor, many enzymes would simply be inactive, or at least significantly less efficient. They wouldn't be able to perform their vital roles.
So the protein alone isn't always enough.
Correct. And there's some specific terminology here that's helpful. When an enzyme is complete and catalytically active with its bound cofactor, It's called a holoenzyme.
Coenzyme, the whole active thing.
Exactly. Think whole. The protein component alone, without its cofactor, is known as an apoenzyme.
A coenzyme, just the protein part.
Right. And in this apoenzyme state, it is typically inactive. This distinction really highlights that the protein part is necessary, but often not sufficient, for full enzymatic function. It's like having a car body without an engine or maybe specific tools.
Got it. So what kinds of cofactors are we talking about here? Are they all the same or do they come in different varieties?
They definitely come in a few distinct varieties, each playing unique roles. One important type is metal ions.
Like minerals.
Exactly. These are inorganic ions like magnesium, zinc, zin, phones, iron, ferro, copper, Cu, manganese, Mu.
Common minerals we hear about.
Right. Enzymes that require these metal ions are often called metalloenzymes. For example, manganese is essential for the activity of arginase, an enzyme involved in the urea cycle. Copper is required for chirosinase, important in making melanin, the pigment in our skin.
What do these metal ions actually do?
They can play several crucial roles. They might help with substrate binding, maybe acting as a bridge between the enzyme and substrate. They can help stabilize the enzyme's delicate three-dimensional structure. Or, very importantly, they can directly participate in redox reactions by accepting or donating electrons, acting like tiny molecular switches.
Versatile little things. What's the other type of cofactor?
The other major type of cofactor is coenzymes.
Coenzymes.
Okay. These are small, organic molecules, not inorganic ions that bind to the enzyme, often transiently or loosely during the reaction cycle.
Organic molecules.
Yes. And they essentially act as carriers. They shuttle specific functional groups or electrons between different molecules during a reaction. They pick something up here, drop it off there.
Like little delivery trucks.
That's a great analogy. And what's truly fascinating and really important for nutrition is that many coenzymes are derivatives of vitamins.
The vitamin connection.
Exactly. This connection is precisely why vitamins are so crucial for our health. Our bodies generally cannot synthesize these vital organic molecules themselves, or not in sufficient amounts. So we must obtain them through our diet to ensure our vast array of enzymes can function correctly. It's a direct link between the food you eat and the molecular machines running your body.
Can you give some examples? Link a vitamin to a coenzyme.
Absolutely. Let's look at some powerful examples. NAD plus IN, or nicotinamide adenine dinucleotide.
Heard of that one.
It's derived from niacin, which is vitamin B3. NAD plus functions as a crucial electron acceptor in many, many redox reactions, like in that lactate dehydrogenase reaction we mentioned. It helps shuttle energy around the cell.
Okay, niacin becomes NAD plus, what else?
Then there's FAD, flavin adenine dinucleotide. This comes from riboflavin, which is vitamin B2. FAD is also heavily involved in redox reactions, particularly in the mitochondria during energy production.
Vitamin B2 gives us FAD. Got it.
Coenzyme A, often written as CoA, is derived from pantothenic acid vitamin B5. Coenzyme A is absolutely critical for the transfer of S-sil groups, which is central to fatty acid metabolism and the citric acid cycle. Think energy metabolism again.
B5 for coenzyme A.
Okay. And even ATP, adenosine triphosphate, which we usually think of as the main energy currency of the cell.
Right, the energy molecule.
It's also often considered a coenzyme, particularly for those lagase reactions, EC6 we talked about. It can donate phosphate groups or simply provide the necessary energy burst needed for joining molecules together.
Interesting. Is there another category here?
Yes, there's a special and very important distinction within coenzymes. Prosthetic groups.
Prosthetic groups? Like artificial limbs?
Not quite. In this context, prosthetic groups are coenzymes that are very tightly, sometimes even covalently, bound to the enzyme.
So they're stuck on there permanently?
Essentially, yes. Unlike other coenzymes that might associate and dissociate bind, do their job, prosthetic groups do not leave the enzyme during the reaction cycle. They are basically a permanent integral part of the active holoenzyme structure.
Okay. Example.
Heme is a classic example. The heme group, which contains iron, found in enzymes like catalase or cytochrome P450 enzymes. That heme is a prosthetic group. It's permanently associated with the enzyme protein, ready to participate in its catalytic function, like breaking down hydrogen peroxide in the case of catalase.
It's incredible how much goes into making an enzyme work. Effectively, its intricate protein structure, these essential cofactors like metal ions and coenzymes. But now that we understand what they are and what they need, what influences how effectively they actually do their job moment by moment? Let's talk about the dynamics of enzyme activity.
Okay, yeah. The factors affecting reaction rate.
First up, substrate concentration. We see that as you increase the amount of substrate, the reaction velocity initially increases rapidly, but then it seems to plateau, it levels off. What's happening at that plateau?
That's a key observation in enzyme kinetics, and it points directly to a fundamental concept, maximal velocity, or
Oh,
Vmax.
maximum speed.
Exactly. At lower substrate concentrations, there are plenty of available active sites on the enzyme molecules. So as you add more substrate, more active sites get occupied, and the reaction speeds up pretty much proportionally. It's like adding more cars to a mostly empty highway traffic flows faster.
Makes sense.
However, at high substrate concentrations, you reach a critical point.
Mm.
All the active sites on all the enzyme molecules become completely saturated. They're all occupied.
The highway's full.
Pretty much. Every enzyme is continuously engaged in binding substrate, converting it to product, releasing the product, and then immediately bonding another substrate molecule. It's working flat out. At this point, the enzyme is operating at its absolute maximum capacity, its Vmax. Adding even more substrate won't increase the reaction rate further. It's simply as fast as that given amount of enzyme can possibly go.
Okay, saturation leads to Vmax. What about temperature? We usually think of heat speeding things up, and that's generally true for chemical reactions, but is there a limit for these delicate biological catalysts?
You're absolutely right to question that, because for enzymes, it's a crucial balance. It's not a simple hotter is always faster situation.
Right.
While increasing temperature generally increases the kinetic energy of molecules, they move faster, collide more often, and with more force, which initially speeds up the enzymatic reaction rate.
As expected.
Enzymes, being proteins, have a very specific optimum temperature.
A sweet spot.
Exactly. For most human enzymes, this optimum is typically around 35 to 40 degrees Celsius, which is right around our normal body temperature. Up to this point, activity increases.
But what happens if it gets hotter than that?
Beyond this optimum temperature, the enzyme's delicate three-dimensional structure begins to unravel. The heat energy becomes too much, breaking the weak bonds holding the protein in its specific shape. This unfolding process is called denaturation.
Denaturation. That sounds bad.
It is. This unfolding is catastrophic for function. It leads to a rapid and often irreversible decrease in activity and a permanent loss of the enzyme's catalytic ability. Think of what happens when you cook an egg white.
It turns solid and white.
Right. The proteins denature, change shape permanently, and you can't uncook it. It's a vivid example of irreversible heat denaturation.
So temperature is a double-edged sword. Too cold, slow reaction. Too hot, destroyed enzyme.
Precisely. Although it's fascinating to note that enzymes from thermophilic bacteria, those organisms living in hot springs, have evolved to have much higher optimal temperature, sometimes up to 70 degrees Celsius or even more. It shows nature's incredible adaptability.
Wow, so temperature can literally cook an enzyme. What about pH? How does the acidity or alkalinity of the environment affect these delicate molecular machines?
pH is another incredibly critical factor, just as vital as temperature. Each enzyme has a specific optimum pH at which its activity is maximal. It operates with peak efficiency only within a narrow pH range.
Another sweet spot.
Exactly. Deviations from this optimal pH, either becoming too acidic or too alkaline, can significantly impact enzyme function.
How does pH affect them?
pH directly alters the ionization state, the electrical charge of the amino acid residues, particularly those found within the enzyme active site.
Changes the charges on the amino acids.
Yes. And changes in the electrical charge of these crucial residues can mess up the enzyme's ability to bind its substrate properly, or it can interfere with the intricate catalytic mechanism itself. Just like with extreme temperatures, at extreme pH values, very acidic or very alkaline, the enzyme can denature completely, leading to an irreversible loss of function.
Can you give examples of different pH optima?
Sure. Consider these contrasts. Pepsin, an enzyme that starts protein digestion in your stomach, functions best in a highly acidic environment, with an optimum pH around 2.
Makes sense for the stomach acid.
Absolutely. In stark contrast, trypsin, which continues protein digestion in your small intestine, prefers a more neutral to slightly alkaline environment with an optimum pH typically between 7 and 9, matching the conditions there.
Different environments, different optimals.
Right. And then you have something like alkaline phosphatase, found in bone, liver, and kidney, which, as the name suggests, thrives in an alkaline environment with an optimum pH around 9 to 10. These examples vividly demonstrate how enzymes are exquisitely tailored, through evolution, to their specific biological locations and roles.
That really illustrates how specialized they are. Okay, this next one seems intuitive, but let's confirm. If you have more workers available, do you generally get more work done, assuming there's enough material? What happens with enzyme concentration?
You're absolutely right. It is intuitive, and the principle holds perfectly true for enzymes. Assuming you have plenty of substrate available so substrate isn't the limiting factor.
Right, assuming Vmax.
Absolutely. This linear relationship is incredibly useful and is often the basis for laboratory enzyme assays. By measuring the reaction rate under conditions where substrate is plentiful, you can determine precisely how much active enzyme is present in a biological sample, like blood or tissue. It's fundamental for quantifying enzyme levels.
Okay. One last factor. What about the product itself? Can the stuff the enzyme makes affect how well the enzyme works?
Yes, it can. We should also consider the impact of product concentration. While maybe not always thought of as a primary regulator, a high concentration of the reaction products can sometimes inhibit enzyme activity.
How?
It can happen in a couple of ways. Sometimes the product might be structurally similar enough to the substrate that it can actually compete for binding at the active site, a form of competitive inhibition.
Yeah, the product gets in the way.
Or, perhaps more commonly, high product levels can subtly shift the reaction equilibrium back towards the reactants, according to Le Chatelier's principle. Basically, if you have a huge pile of product building up, it makes it thermodynamically less favorable for the forward reaction to keep going.
Okay.
And interestingly, this accumulation of product can serve as a form of feedback regulation. High product levels can signal back to the enzyme, effectively saying, okay, we've made enough for now, helping to prevent wasteful overproduction. It's another layer of the cell's smart control system.
We've explored the individual knobs and dials controlling enzyme activity, substrate, temperature, pH, enzyme concentration, even product buildup. But observing isn't always enough for scientists. We need to quantify it, predict it. And that brings us to the mathematical heart of enzyme function, kinetics. Let's delve into enzyme kinetics, starting with the Michaelis-Menten model. This is a cornerstone, right? How does it describe reaction velocity and help us understand enzyme behavior?
Yes, the Michaelis-Menten equation is absolutely a cornerstone of enzyme kinetics. It provides a fundamental mathematical description of how the reaction velocity, or rate, varies with the concentration of the substrate.
Okay, a mathematical model.
It's a beautifully elegant and relatively simple model that, at its core, makes a few key simplifying assumptions. It typically assumes there's only a single substrate interacting with the enzyme, that each enzyme molecule has a single active site, and very importantly, that the reaction quickly reaches a steady state.
Steady state. What does that mean here?
Steady state in enzyme kinetics means that the concentration of that enzyme substrate, ES complex we talked about, remains relatively constant over the time period we're measuring.
So the amount of enzyme currently bound to substrate doesn't fluctuate wildly.
Exactly. The rate at which the ES complex is forming, enzyme binding substrate, is balanced by the rate at which it's breaking down, either releasing substrate again or converting substrate to product. Imagine that busy factory assembly line again. Raw materials are constantly coming in, finished products are going out, but the number of items actively being worked on at any given moment stays roughly the same. It's a dynamic balance, crucial for making reliable measurements.
And when we measure the rate, we look at the beginning.
Yes, typically we measure the initial velocity, vital year. That's the rate of product formation measured right at the very beginning of the reaction, usually within the first few seconds or minutes.
Why the beginning?
We do that before significant amounts of product have accumulated, which could cause that product inhibition we just discussed, and before the reverse reaction, product turning back into substrate, becomes significant, which can happen as product builds up. Measuring VO helps us isolate the enzyme's intrinsic catalytic efficiency under defined conditions.
Okay, so at the heart of the Michaelis-Menten model are two critical values, Vmax, which we've discussed, and this other term, KOM. Can you elaborate on these two? What do they tell us beyond just being numbers on a graph? What's their real significance?
Absolutely. They're the key parameters. Vmax, as we covered, is the maximal velocity or rate the enzyme can achieve when it is completely saturated with substrate. It's a direct reflection of the enzyme's turnover number, its inherent catalytic power, how quickly it can convert substrate into product when substrate availability isn't limiting. A higher Vmax generally means a faster or more efficient enzyme per molecule.
Got it. Vmax is the speed limit. Now, what about Km?
Klm, the Michaelis constant, is a truly pivotal value. You can almost think of it as part of the enzyme's personality profile regarding its substrate. Klm represents the substrate concentration at which the reaction velocity, dTA, is exactly half of Vmax.
Half the maximum speed. Okay, why is that concentration important?
It's important because it's a critical indicator of the enzyme's affinity for its substrate, how sticky the enzyme is for its substrate.
Affinity, how tightly it binds.
Exactly. A small kilometer value indicates that the enzyme has a high affinity for its substrate. It takes only a small amount of substrate to get the enzyme working at half its maximum speed, implying tight binding. The enzyme gets saturated easily.
Low coulomb, high affinity. Makes sense.
Conversely, a large kilometer value indicates a low affinity. This means a higher concentration of substrate is needed to reach half of Vmax, suggesting weaker binding. The enzyme needs a lot more substrate around to get close to its maximum speed.
High coulomb, low affinity.
Right. So coulomb reflects how tightly the enzyme binds its substrate and is a characteristic constant value for any given enzyme-substrate pair under specific conditions, like temperature and pH. It provides crucial insights into their interaction. Drug designers, for instance, pay very close attention to coulomb values and how potential drugs might affect them. It tells them how well their drug might compete with the natural substrate.
So coulomb tells you about binding, Vmax tells you about catalytic speed.
That's a good summary, yes. They describe different aspects of the enzyme's function.
The idea of quantifying enzyme speed with a curve is powerful, but you said the Michaelis-Menten curve is hyperbolic. I can imagine it being tricky to pinpoint Vmax exactly, since the curve just sort of approaches it asymptotically, and getting Km, half Vmax, precisely from that curve might also be difficult.
You've hit on a key practical challenge. Reading precise values, especially Vmax, directly from a hyperbolic curve can indeed be inaccurate. It's hard to know exactly where that plateau is.
So are there any clever mathematical tricks scientists use to make that data easier to work with and get more accurate numbers?
There is. A very widely used graphical method is the Lineweaver-Burke plot, which is also known as the double reciprocal plot.
Double reciprocal. Sounds mathematical.
It is, but the concept is straightforward.
Yeah.
You take the reciprocal of both the reaction velocity, so 1 VBO, and the reciprocal of the substrate concentration, 1S. Then you plot 1 V0 on the y-axis against 1S on the x-axis.
So you're plotting the inverses. Why?
The beauty of this mathematical transformation is that it converts the hyperbolic Michaelis-Menten relationship into a straight line.
Ah, straight lines are much easier to analyze.
Exactly. Linear relationships are much easier to extrapolate and interpret visually. This linearization offers several significant benefits.
Like what?
Well, you can easily and accurately determine Vmax by looking at where the straight line intercepts the y-axis. The y-intercept value is actually equal to 1 Vmax.
Okay, so flip the y-intercept, get Vmax.
Right. Similarly, Kelm can be determined from the x-axis intercept. The point where the line crosses the x-axis corresponds to -1 kilometer.
Flip the negative x-intercept to kilometer.
Precisely. Beyond just determining these kinetic parameters with greater precision, the Lineweaver-Burke plot is also incredibly valuable for analyzing enzyme inhibition.
How so?
Different types of inhibitors, which we'll get into, produce distinct and predictable changes in the slope and intercepts of the Lineweaver-Burke plot. This makes it an indispensable tool for figuring out how an inhibitor is working, whether it's competing with the substrate, binding elsewhere, etc. It's a brilliant piece of kinetic analysis, really powerful for research and drug development.
Okay, so enzymes are these powerful catalysts, finely tuned, quantified by kinetics. But their activity isn't always just running at full tilt or depending only on substrate levels. Our bodies need robust ways to control them, to turn them up or down, or even to stop them completely when needed. And enzyme inhibition sounds like a major way this critical control happens. How do we classify these molecular breaks?
Enzyme inhibition is indeed a critical regulatory mechanism, absolutely essential for cellular control. And yes, we classify inhibitors based primarily on how they interact with the enzyme and whether that interaction is permanent or temporary. The two main categories are reversible and irreversible inhibition.
Okay, let's start with reversible. Sounds like it's temporary.
Exactly. In reversible inhibition, the inhibitors bind non-competently to the enzyme using weaker interactions like hydrogen bonds or ionic bonds. This means the binding is temporary. The inhibitor can bind, and then it can dissociate, leaving the enzyme active again. It's like hitting a pause button, not the stop button.
Makes sense. Are there different kinds of reversible inhibition?
Yes, there are three main types, and they differ in where the inhibitor binds and how that affects the enzyme's kinetics, particularly Vmax and chimerism.
Okay, what's the first type?
First, we have competitive inhibition. In this scenario, the inhibitor molecule often looks structurally very similar to the enzyme's natural substrate.
A look-alike molecule.
Exactly. Because it looks similar, it can bind to the enzyme's active site, the same spot the substrate wants to bind to. So it literally competes with the substrate for access to the active site. Think of it like a game of molecular musical chairs.
Okay, they fight for the same seat. How does that affect Vmax and Caremo?
Because the inhibitor and substrate are competing for the same site, if you add enough substrate, really flood the system with it, you can eventually outcompete the inhibitor and the enzyme can still reach its normal Vmax. The maximum speed isn't fundamentally changed, you just need more substrate to get there.
So Vmax stays the same. What about Caremo?
Ah, but because you need more substrate to reach half Vmax in the presence of the competitor, the apparent kilomem increases. It looks like the enzyme has a lower affinity for its substrate when the inhibitor is around.
Increased kilom, same Vmax. Got it. Is this used in medicine?
Immensely. Competitive inhibition is a cornerstone of drug design. A classic, crucial example is the action of statins, the cholesterol-lowering drugs like atorvastatin or pravastatin.
Right, lots of people take those.
They are designed to look like a natural intermediate in the cholesterol synthesis pathway and competitively inhibit a key enzyme called HMG-CoA reductase. By blocking this enzyme's active site, statins effectively reduce the body's production of cholesterol. It's a perfect example of targeted competitive inhibition saving lives.
Wow, okay, what's the next type of reversible inhibition?
Second, we have non-competitive inhibition.
Yeah.
Here, the inhibitor binds reversibly, but not at the active site. It binds to a different distinct site on the enzyme.
Somewhere else on the enzyme molecule.
Yes. And this binding, even though it's not at the active site, causes a conformational change in the enzyme. It subtly warps the enzyme's overall shape, including the active site, just enough to reduce its catalytic efficiency.
So it makes the enzyme work slower, even if the substrate can still bind.
Exactly. A key feature is that a non-competitive inhibitor can often bind to both the free enzyme and the enzyme substrate ES complex. It doesn't care if the substrate is already there or not.
Okay, how does that affect Vmax and Caremo?
Because the inhibitor reduces the catalytic efficiency of the enzyme molecules it binds to, the overall Vmax decreases. The enzyme simply can't work as fast no matter how much substrate you add. However, because the inhibitor doesn't block the substrate from binding to the active site, it binds elsewhere. It doesn't affect the enzyme's affinity for the substrate, so the kilomem remains unchanged.
Decreased Vmax, same kilomem. Interesting. An example.
An example often cited is heavy metal ions like lead or mercury, though those are often more complex or irreversible. A pharmacological example could be something like D-allopurinol, a metabolite of the gout drug allopurinol, which can act as a non-competitive inhibitor of xanthine oxidase, an enzyme involved in producing uric acid.
Okay. And the third type of reversible?
Third, and a bit more unique, is uncompetitive inhibition. This is where the inhibitor binds only to the enzyme substrate ES complex. It cannot bind to the free enzyme.
Only after the substrate is already bound. That sounds sneaky.
It is a bit different. It essentially waits for the enzyme to grab the substrate, and then it binds, effectively locking the substrate in place or preventing the product from being released properly. It stabilizes the ES complex, but in a non-productive way.
So it traps the enzyme in the middle of its job. What does that do to Vmax and Caremo?
This leads to a unique kinetic signature. Both Vmax and kilomem decrease, typically proportionally. Vmax decreases because the inhibitor is preventing the catalytic cycle from completing efficiently. Kilomem decreases because, by binding only to the ES complex, the inhibitor actually pulls the equilibrium towards forming more ES complex, making it look like the enzyme has a higher affinity for the substrate, hence lower kilom, even though it's an inhibitory effect overall.
Both Vmax and kilomem go down. Okay, that's distinct. Now, what about the other main category, irreversible inhibition? Sounds permanent.
It generally is. In irreversible inhibition, the inhibitor forms a very stable, often covalent bond with the enzyme, usually targeting a key amino acid residue within the active site. Or it might form an extremely tight, non-covalent complex that essentially never dissociates. This permanently inactivates the enzyme molecule.
Like breaking the key off in the lock.
That's a very good analogy. These are often referred to as suicide inhibitors, because the enzyme itself often participates in its own inactivation. It might start to process the inhibitor as if it were a substrate, but in doing so, it converts the inhibitor into a highly reactive form that then permanently attaches to the enzyme.
The enzyme triggers its own demise. Clever. Are these used as drugs too?
Oh, absolutely. Irreversible inhibitors are incredibly important clinically. Consider aspirin. It irreversibly inhibits cyclooxygenase, COX enzymes, by covalently attaching an acetyl group to a serine residue in their active site. This blocks the production of prostaglandins, which are involved in inflammation, pain, and fever.
So aspirin permanently shuts down those enzyme molecules.
The ones it binds to, yes. The body has to synthesize new enzyme molecules to restore activity. Similarly, many vital antibiotics like penicillin and amoxicillin are suicide inhibitors. They target enzymes called transpeptidases that bacteria need to build their cell walls. The enzyme starts to process the penicillin, gets stuck, and the bacterium can't build its wall properly, eventually dying.
Wow. Any other examples?
ACE inhibitors, the blood pressure medications like captopril and enalapril lisinopril, they are potent inhibitors, often considered effectively irreversible, or very slowly reversible, of angiotensin-converting enzyme, a key player in regulating blood pressure. By blocking this enzyme, they help relax blood vessels and lower pressure. The specificity and potency of these inhibitors, whether reversible or irreversible, are critical considerations in drug design. You want to hit your target enzyme strongly without causing unwanted side effects by inhibiting other crucial enzymes.
It's amazing how precisely we can target these enzymes with inhibitors. But inhibition isn't the only way to control them, right? You mentioned enzymes responding to signals at sites other than the active site. That sounds like allosteric regulation, like a remote control system.
That's a perfect analogy.
Yeah.
Allosteric regulation is a major and incredibly sophisticated way cells fine-tune metabolic pathways. Allosteric enzymes are typically larger, often multi-subunit enzymes, meaning they're built from multiple protein chains, and they usually possess multiple active sites.
Multiple subunits, multiple active sites. Okay.
What truly defines them is their ability to respond to the binding of specific regulatory molecules, called effectors or modulators, at sites that are distinct and physically separate from the active site. These regulatory binding spots are known as allosteric sites, meaning other shape or other site.
So the control molecule binds somewhere else entirely.
Exactly. And the binding of an effector at an allosteric site induces a conformational change that ripples through the enzyme's structure, ultimately affecting the shape and function of the active sites, even if they're quite far away physically on the molecule. It's like flipping a switch on one side of the machine affects the tools on the other side.
A cellular communication network within the enzyme. What kinds of effects can these molecules have?
These effectors can be either positive effectors, also known as activators, which increase enzyme activity. They often do this by making it easier for the substrate to bind, effectively decreasing the apparent Keller matter, or K0.5, as it's often called for allosteric enzymes, or by increasing the enzyme's maximum catalytic rate, increasing Vmax.
Turning the enzyme up.
Or they can be negative effectors, also known as inhibitors, which decrease enzyme activity. They typically work by making substrate binding harder, increasing K0.5, or by lowering Vmax.
Turning the enzyme down.
Right. It allows for very sensitive tuning of enzyme activity based on the cell's current needs, as reflected by the concentrations of these effector molecules.
You mentioned a different term there, K0.5 instead of kilome. And does allosteric regulation affect the kinetics plots we talked about?
Good catch. Yes, the kinetics of allosteric enzymes are often different from the simple Michaelis-Menten enzymes. Instead of the hyperbolic curve, allosteric enzymes frequently display sigmoidal, S-shaped kinetic curves when you plot reaction velocity versus substrate concentration.
S-shaped, not hyperbolic. Why?
This sigmoidal shape is a hallmark of cooperative binding. It means the binding of one substrate molecule to an active site on one subunit can actually influence the binding of subsequent substrate molecules to the other active sites on the same enzyme, often making that subsequent binding easier.
Like the first one primes the bump.
Exactly. It's like the enzyme gets warmed up or becomes more receptive once it starts binding substrate. This allows for a much more sensitive response to small changes in substrate concentration, especially around the K0.5 value, which is analogous to kilominute, the substrate concentration needed for half maximal velocity, but used for sigmoidal curves. A small increase in substrate can cause a large jump in activity.
Is hemoglobin an example of this, even though it's not an enzyme?
It's the classic textbook example of cooperativity. Oxygen binding to one heme group in hemoglobin increases the affinity of the other heme groups for oxygen, leading to that characteristic S-shaped oxygen binding curve. It allows blood to efficiently load up with oxygen in the lungs where it's plentiful and efficiently release it in the tissues where it's less concentrated. Allosteric enzymes use the same principle for metabolic regulation.
Okay. And these effectors, the molecules binding to the allosteric site, can the substrate itself be an effector?
Yes, it can. We distinguish between two types based on the effector molecule. If the substrate itself acts as an allosteric effector, usually a positive one, enhancing its own binding or the binding of more substrate, it's called homotropic regulation. Cooperativity in substrate binding is a form of homotropic regulation.
Substrate affects its own binding. Got it.
If the effector is a molecule different from the substrate, it's called heterotropic regulation. This is where those activators and inhibitors we mentioned come in molecules that signal the cell's metabolic state.
Like the end product of a pathway.
Exactly. A very common and elegant example of heterotropic regulation is feedback inhibition. This is where the final end product of a multi-step metabolic pathway binds to an allosteric site on an enzyme that acts early in that same pathway, inhibiting its activity.
So when you have enough product, it shuts down its own production line at the start.
Precisely. It prevents the cell from wasting energy and resources, making something it already has in sufficient supply. It's an incredibly efficient and common biological control loop.
Beyond these more immediate dynamic controls like inhibition and allosteric effects, are there longer-term ways the body adjusts enzyme levels and activity, maybe over hours or even days?
Yes, absolutely. Cells have mechanisms for longer-term adaptation as well. Two major strategies here are covalent modification and controlling enzyme synthesis.
Okay, let's start with covalent modification. What does that involve?
Covalent modification means regulating an enzyme's activity by chemically attaching or removing specific chemical groups directly onto the enzyme protein itself, using covalent bonds.
Adding or removing tags.
Sort of, yes. The most common type by far is phosphorylation and dephosphorylation.
Adding phosphates.
Exactly. Enzymes called kinases add phosphate groups, usually taking them from ATP, onto specific amino acid residues like serine, threonine, or tyrosine on the target enzyme. And other enzymes called phosphatases remove these phosphate groups.
Kinases add, phosphatases remove. Okay. What does adding a phosphate do?
Adding that bulky, negatively charged phosphate group can cause a significant conformational change in the enzyme, which can either activate it or inactivate it, depending on the specific enzyme. It's like flipping a molecular switch. For instance, glycogen phosphorylase, the enzyme that starts breaking down stored glycogen into glucose, is activated by phosphorylation when your body needs quick energy. This provides a rapid on-off switch that can integrate with cellular signaling pathways.
So it's another layer of control, often linked to hormones or signals.
Very often, yes. It's faster than changing the amount of enzyme, but often slower than allosteric effects.
What about changing the amount of enzyme? You mentioned synthesis control.
Right. This involves regulating enzyme activity at the genetic level, controlling how much of the enzyme protein is actually made in the first place. This is done through induction and repression of gene expression.
Induction and repression.
Yeah.
Turning gene activity up or down.
Exactly. The body can increase or induce the synthesis of specific enzyme molecules when more of that enzyme's activity is needed over the long term. Or it can decrease or repress their synthesis when less activity is required.
So you're changing the number of enzyme molecules in the cell.
Precisely. This fundamentally alters the total catalytic capacity for that reaction. Because it involves changes in gene expression and protein synthesis, this is a much slower, more gradual regulatory mechanism compared to the near instantaneous effects of allosteric regulation or the relatively rapid covalent modifications. It's how your body adapts to sustained changes, like long-term dietary shifts or chronic exposure to certain drugs or toxins.
Makes sense for long-term adaptation. Is there anything else maybe broader?
One final, very important regulatory strategy is compartmentalization.
Compartmentalization, keeping things in different places.
Exactly. Cells are highly organized internally, with various organelles like the mitochondria, nucleus, lysosome, cytosol, etc. Enzymes and their substrates are often physically segregated into specific compartments.
Why is that helpful?
It offers several advantages. It localizes specific metabolic pathways to particular locations, preventing interference between potentially competing or incompatible reactions. For example, the enzymes for building fatty acids synthesis are mainly located in the cytosol,
Okay.
while the enzymes for breaking down fatty acids for energy, oxidation, are primarily found inside the mitochondria. This physical separation prevents a futile cycle where synthesis and breakdown happen simultaneously. It also allows the cell to maintain different biochemical environments like pH or cofactor concentrations, optimized for specific sets of reactions within each compartment. It's an organizational masterpiece underpinning cellular efficiency.
It's clear that enzymes are absolutely essential for life's processes within the cell, intricate, powerful, and exquisitely regulated. But how does all this intricate knowledge extend into the real world, particularly in clinical settings and medical applications? Enzymes aren't just workhorses inside our cells, they're also crucial signals and targets for what's happening in our bodies, aren't they?
They most certainly are. Enzymes play hugely important roles, both as diagnostic tools and as therapeutic targets.
Okay, let's talk diagnostics first. How do enzymes give us clues about health?
Well, normally, most enzymes do their work inside cells. So you usually find only very small baseline amounts of these intracellular enzymes circulating in the plasma, the liquid part of blood.
Right, they should stay inside the cells mostly.
Exactly. However, if tissues are damaged, say, due to injury, inflammation, infection, or lack of oxygen, the cell membranes can become leaky or rupture entirely. When this happens, the intracellular enzymes spill out from the damaged cells into the bloodstream.
So high levels in the blood mean cell damage somewhere.
Precisely. Measuring significantly increased plasma levels of specific intracellular enzymes often serves as a critical indicator of tissue damage or disease. It tells doctors that something is wrong, and often where something is wrong.
How do you know where the damage is?
That's where isoenzymes become incredibly useful. Sometimes they're called isozymes.
Isoenzymes. What are those?
These are different forms of the same enzyme. They catalyze the exact same chemical reaction, but they have slightly different amino acid sequences due to being coded by different genes, or sometimes due to modifications after they're made. These structural differences give them distinct physical properties, like how they move in an electric field, electrophoretic mobility, or how they respond to inhibitors.
Different versions of the same enzyme.
Okay, and crucially, different tissues often produce different isoenzyme forms, or different proportions of isoenzymes. So by measuring the specific pattern of isoenzymes present in the plasma, doctors can often pinpoint the location and sometimes even the nature of the tissue damage much more accurately. They're invaluable diagnostic markers.
That sounds incredibly precise. Can you give us a vivid example, something common where isoenzymes are crucial?
A prime life-saving clinical example is the diagnosis of a myocardial infarction, or MI, what most people call a heart attack.
Okay, heart attacks, very important.
Here, measuring a few key isoenzymes in specific cardiac proteins in the blood is absolutely routine and critical for diagnosis and management.
Which ones?
First, there's creatine kinase, or CK, sometimes called CPK. CK exists in three main isoenzyme forms. CKMM, which is predominantly found in skeletal muscle.
Like from exercise.
Right. Then there's CKBB, found primarily in the brain, and importantly, CKMB, which is highly enriched in cardiac muscle in the heart.
CKMB for heart muscle.
Exactly. So finding significantly elevated levels of CKMB in the plasma is a strong and relatively specific indicator of damage to the heart muscle consistent with an MI. Its levels typically start to rise 4 to 8 hours after the onset of chest pain, peak around 24 hours, and then return to baseline within about 2 to 3 days. The specific timing and the height of the peak give doctors important information.
Okay, CKMB is a key heart attack marker. Is that the main one used now?
It was historically very important and still measured, but the gold standard biomarkers for MI diagnosis today are actually cardiac troponins, specifically CTNI and CTNT.
Troponins? Are they enzymes?
No, they're actually structural proteins, part of the contractile machinery of heart muscle cells. But they are released into the blood when heart muscle is damaged, just like enzymes.
Why are they the gold standard?
They offer even higher specificity and sensitivity for detecting heart damage than CKMB. They are virtually only found in heart muscle, so their presence in the blood is a very reliable sign of MI. They also start to rise around the same time as CKMB, 4 to 6 hours post to MI, but crucially, they remain elevated for much longer, several days, sometimes up to a week or two.
So they give a longer window for diagnosis.
Exactly. They can help detect heart attacks even if the person seeks medical attention a bit later, and they can also help detect smaller amounts of heart damage.
Any other enzymes used?
Another enzyme sometimes monitored, though less specific than troponins, is lactate dehydrogenase, or LDH. We mentioned LDH earlier as an oxidoreductase. It has 5 common isoenzymes, LDH1 through LDH5, each with different tissue distributions. LDH1 is highest in cardiac muscle and also red blood cells. So an increase in plasma LDH1 relative to LDH2, which is normally higher in plasma, can also suggest MI, particularly if the LDH flip pattern is seen, but it's used less now with the superiority of troponins.
So the patterns and timing are key.
Absolutely critical. Analyzing the rise and fall patterns of these specific markers, CKMB, and especially troponins, allows doctors to accurately diagnose MI, estimate the extent of the damage, monitor the patient's progress, and make crucial decisions about treatment, like whether to use clot-busting drugs or perform procedures like angioplasty. It's a remarkable example of using biochemistry for critical care.
So enzymes help us diagnose diseases, providing these vital biochemical clues, but they're also frequently the direct targets of drugs, acting as the very switches that doctors want to turn on or, more often, turn off, aren't they?
Yes, absolutely. Beyond diagnostics, enzymes are arguably one of the most important classes of drug targets. Many pharmaceutical drugs are meticulously designed either to inhibit or, much less commonly, to activate specific enzymes in order to treat a wide range of diseases.
Why are they such good targets?
Because they often play critical, rate-limiting roles in essential biochemical pathways. By precisely targeting a specific enzyme with a drug, you can directly intervene in a pathway that might be overactive, like cholesterol synthesis, or producing harmful products, like prostaglandins in inflammation, or perhaps one that's essential for the survival of a pathogen, like bacterial cell wall synthesis. It's about restoring biochemical balance or selectively disrupting a disease process.
We've touched on some examples, but maybe recap a few key areas where enzyme-targeted drugs are vital.
Sure. We mentioned antibiotics like penicillin and amoxicillin, which are lifesavers because they irreversibly inhibit bacterial transpeptidases needed for cell wall synthesis. Bacteria have these enzymes, we don't, making them excellent selective targets.
Right, targeting the pathogen.
We talked about antihypertensives, like the ACE inhibitors,
Yeah.
captopril and allopril, etc., which block the angiotensin-converting enzyme to lower blood pressure. That's targeting a key enzyme in our own physiology that's become dysregulated.
And managing our own systems.
And in chemotherapy, many anti-cancer drugs work by targeting enzymes that are essential for the rapid growth and DNA replication of cancer cells. For example, drugs like methotrexate inhibit dihydrofolate reductase, an enzyme needed for making DNA precursors, thus slowing down cancer cell proliferation more than normal cells.
Targeting runaway growth.
Exactly. Statins for cholesterol, aspirin for inflammation and clotting, drugs for HIV that inhibit viral enzymes like reverse transcriptase or protease. The list is enormous. Understanding enzyme structure, mechanism, and kinetics is absolutely fundamental to modern drug discovery and development.
And are we getting better at designing these drugs, maybe even engineering enzymes themselves?
Yes, absolutely. The field is constantly advancing. And on an even more exciting front, there's the rapidly growing field of enzyme engineering.
Engineering enzymes, modifying them.
Precisely. Using tools like genetic engineering and directed evolution, scientists can now modify natural enzymes or even design new ones from scratch to improve their properties for specific applications.
Like what kind of improvements?
They might engineer an enzyme to be more stable at high temperatures or in harsh chemical conditions, making it more suitable for industrial processes like in detergents or food production. They might alter its specificity to make it react only with the desired target molecule, reducing side reactions. Or they could enhance its catalytic activity to make it work much faster.
What about therapeutic uses for engineered enzymes?
That's a huge area of potential, too. Imagine enzymes designed for targeted drug delivery, perhaps activating a prodrug only at a tumor site, or enzymes developed to break down specific toxins or pollutants in the environment or even within the body. There's research into using engineered enzymes to generate novel biofuels more efficiently. The possibilities of harnessing and improving upon nature's catalysts are truly expanding at an incredible pace.
What an incredible, profound journey we've taken today. We've gone from defining enzymes as life's indispensable microscopic catalysts.
The biological workhorses.
Well, exactly. To understanding their intricate classification system, the EC numbers, the 6 main classes. We explored the lock and key and the induced fit precision of their active sites.
Like dynamic interaction.
The crucial role of cofactors, like vitamins and metals that give them that powerful boost. We looked at the kinetic dance dictated by substrate, temperature, pH, and the enzyme's own concentration, quantified by Vmax and CREAM.
And visualized with Lineweaver-Burke plots.
Right. And then we dove into the sophisticated ways they're regulated inhibition, competitive, non-competitive, uncompetitive, irreversible, allosteric control with those remote switches, covalent modification like phosphorylation, and even controlling their synthesis.
All those layers of control.
And finally, how this knowledge translates directly into medicine, using enzymes as diagnostic markers for conditions like heart attacks, and designing drugs that target enzymes to treat countless diseases. It's truly a marvel of molecular engineering occurring right within our own bodies every single second.
It really is. And it's a powerful reminder that every breath you take, every thought you have, every beat of your heart, every single move you make is utterly dependent on the tireless, precise work of these countless enzymes. You are, quite literally, a walking, talking biochemical marvel, powered by these tiny catalysts operating in perfect, intricate harmony. That profound appreciation for these molecular workhorses is absolutely well-deserved when you stop and think about it.
Absolutely. So given their incredible precision, their efficiency, their adaptability, and how much we're learning about them every day, especially with enzyme engineering, what breakthroughs might we see next? as scientists learn to program enzymes for even more complex tasks.
The potential seems vast.
Could we see things like self-repairing biological systems using engineered enzymes to fix damage at a cellular level, or maybe novel energy solutions that mimic the stunning efficiency of photosynthesis or metabolic pathways? How far can we truly push the boundaries of these molecular machines? And what new horizons will they unlock for human health, for industry, for the environment? What stands out to you about the sheer, almost unfathomable power and potential of enzymes as we look forward?